validation. My name is Sarah Crowell, and I'm the Senior Product Manager at Steris Isometric Services. I'll be the host for today's event. Dr. Jerry McDonald and Beth Jack are the presenters for today's webinar. Dr. Gerald McDonald is a Vice President, Research and Clinical Services for Steris Corporation, as well as a consultant with Biotest Laboratories in the design, development, and testing of reusable devices and their instructions for use. Dr. McDonald holds a BSc with honors in Medical Laboratory Sciences and a PhD in microbiology. He is a frequent presenter on infection prevention with over 160 publications and patents in the area. He's an assistant editor for the Journal of Hospital Infection and a reviewer for a number of other leading journals on infection prevention and control topics. Dr. McDonald is a member of national and international working groups, including AMI, ISO, and EN committees for standard and guideline development on areas including sterilization, disinfection, cleaning, and aseptic processing. Beth Jack joined Biotest Laboratories in 2005 as a regulatory and compliance specialist and is now manager of compliance. Ms. Jack has a BSc in biotechnology, chemistry, and microbiology, and has held various laboratory, quality, and regulatory positions, attributing to her more than 20 years of experience. Her expertise has led to the launch of the Reprocessing of Reusable Medical Devices initiative at Biotest Laboratories, and she is involved in the AMI Standards Organization in the areas of microbial management, human factors for device reprocessing, and steam sterilization in healthcare facilities. We're excited that we have a lot of great material to cover in today's webinar. We will include a brief question and answer session at the end of the presentation and would like to encourage everyone to submit questions at any time during the webinar. Once the presentation is completed, Jerry and Beth will answer the questions in the order they were received. You can submit your questions by typing them in the questions field on your GoToWebinar control panel. If you have a question that we don't answer in the time allotted, we will address it separately in an email following the webinar. Today's presentation will be recorded, and we will email a link to download the file to all participants tomorrow. Please note that continuing education credits are not provided as part of this webinar. And now I'd like to turn the phone over to Jerry to begin the presentation.
Well, hello everyone, and thank you very much for joining us today for what we hope would be a brief introduction to some of the details behind reusable devices and their validation requirements with a specific emphasis on processing or reprocessing. Devices, as you can see on this slide, are used for a variety of medical applications, and they range in their specific use and indeed complexity. But for the purpose of today, we will focus on those devices that are particularly designed to be reused. So let's define what we mean by this. So many of you may be familiar with the term single-use device, which is defined as a device intended to be used on a single patient during a procedure and then disposed of. While in contrast to these are the reusable medical devices, or multiple-use devices. And these are devices that are designated or intended by the manufacturer as being suitable for processing and then reuse on another patient. Now, under this definition, we can also consider two separate types of devices. These can include reusable devices that have a defined and often short limit in the number of times that they are reused, such as 5 times, 10 times, or 20 times, to those that can be reused and can be sometimes reused over many years. So it may include hundreds or thousands of times. When the device is used on one person and is being prepared for use on another, it is important to ensure that it is rendered safe for use. So before we proceed into this, it's useful maybe to understand that this can include multiple defined steps that we refer to as processing or reprocessing. And I've given some of the more important definitions in this area for you on this slide. So first of all, reprocessing or processing, and you will hear us use those two terms interchangeably today to mean the same thing, is a process that's carried out on a device in order to allow for its subsequent safe use. And this can include various different steps such as cleaning, disinfection, sterilization, and related procedures. Decontamination is defined as the physical or chemical means to render a surface or item safe for use, handling, or disposal. And this may include many steps also, but it's generally a two-step process. The first step in this is, of course, cleaning. And cleaning, by definition, is the removal of contamination, often referred to as soil, which can include a variety of types of tissues or contamination that can remain on the device after it has been used on a patient, from the surface to the extent necessary for processing. And that will include then disinfection or sterilization, or in certain cases, cleaning may be the only step that's actually done prior to the intended use of the device. Then we move into the antimicrobial processes, and these will include disinfection and sterilization. Disinfection is by definition the antimicrobial reduction of the number of viable microorganisms on a surface to a level previously specified as being appropriate for its intended use or further handling. And that's something that we will go into in a little bit more detail later in the presentation. Sterilization is different. So while disinfection reduces the number of viable microorganisms, sterilization is a defined process used to render a surface or product free from viable microorganisms, including some of the more resistant forms of microorganisms, being bacterial spores. This slide gives you a summary of the typical cycle of reprocessing that can be done in a facility on these types of devices. But different devices can be subjected to different parts of the cycle depending on their risk to a patient or according to the healthcare facility's policies. So you'll see, for example, new devices coming in, and some of them are used once and then discarded immediately. Others are coming in, they're being cleaned, and immediately after being cleaned, they are returned to storage or patient use. Other types of devices then, because of their risk, really need to be subjected to a disinfection or sterilization step before they're used on a patient. And then finally, other devices, in particular critical or surgical devices in many cases, go through the full cycle of cleaning, some sort of disinfection to render them handling or safe for handling, packaging, sterilization, and then sterile storage before patient use. Now this can be quite complicated. It looks very nice on the screen, but you can imagine that a facility with thousands of devices being used potentially on a daily basis, that things get very busy in some of these departments that do this job on a daily basis. Now this is an old slide, and it's a picture of a surgical suite taken from the 1900s. And in many ways things have changed, but in many ways they haven't. So you'll still see the surgery area, for example, with the older surgical light. I'm sure that our technology has improved now, and the surgical tables. But you will also see that that's the center where the patient is being handled, or the patient's surgical procedure is being done. But if you note, in the far corner, there is a designated reprocessing area. In particular, a lot of this was done manually, so you can see one nurse in particular in the corner that was responsible for this. Now, you can expect to see a much more modern approach to this picture in most healthcare facilities today, but device reprocessing can take place in many areas around the facility, sometimes in areas that you would least expect. Now, these are not good examples. Oftentimes, these areas are not fit for purpose. So here you have two cupboards essentially shown in these pictures where you can see that reprocessing is actually happening. And in particular, you have a small sink on the right, the sterilizer sitting beside them, and then waste sitting under them. Not very good practice. Now, these sorts of examples are not acceptable to meet the various standards and guidelines that healthcare facilities are being required to do today. But in particular, these areas are becoming unacceptable with the greater emphasis on the safe processing of devices. But I do show them to you to highlight to you some of the concerns and difficulty that can be in place in different facilities regarding reprocessing of devices and the importance for having clear instructions on how to do it to limit risk. Now here's a much better picture of a well-designed processing department, at least in simulation. And this will include, if we go from the lower left part of the screen, of the delivery of the devices into an area designated as we would call a dirty area, where the devices can be manually sorted and cleaned, and then pass through a wash disinfector in the center to the clean area on the right. This allows for separation of dirty and clean instruments, so devices that could have a bloodborne pathogen risk on the left, to devices that can be safe to be handled on the right. The devices are then sorted into their respective sets in preparation for the surgical procedure, and then prepared for by packaging and then sterilization, using various different types of technologies, such as steam and hydrogen peroxide. And then as we move up the slide, the devices are then passed through to a storage or distribution area in the upper right. Just to give you some practical examples of what these areas look like, this is a typical area for cleaning, with an emphasis on manual cleaning. So for example, you will see two sinks. One is used for cleaning the device, where you might soak in a chemistry for a certain amount of time, and the other is used for rinsing the device. Often an underestimated step, the rinsing of the device, to make sure that there are no toxic residuals that remain on the device prior to a terminal disinfection or sterilization process. You will also see some other types of equipment which are quite typically used in these areas, such as ultrasonic machines that can be used to be able to clean some more difficult to clean devices with ultrasonic technology. Here you have a view of the washer disinfectors. So these are double door washer disinfectors, so you see the dirty side of these on the left, and then you see the clean side on the right. And I think many of you may agree that many of these departments don't look very busy, but that's because many of these pictures are taken before these departments have opened, or where all the staff are not actually there. So they're a little bit too perfect to be shown. But they give you a good idea of the setup of these types of departments. This is a view then of towards the sterilizers. So you will see various different packs of devices with a tracking system, and then also the sterilizers in the distance. In this case, these are steam sterilizers. Now, this is more the reality of we see coming down into these departments on a daily basis. And this slide does show some challenges that these departments face on receiving sets of instruments from reprocessing. So you will see, for example, that there are often patient tissues and blood which has been allowed to dry on the devices, immediately making the device more difficult to reprocess. You will also see safety risks from sharps or other types of instruments which could cut into the gloves of people and maybe even into their skin, which is, of course, a risk to their health. You will also note that for many of those devices that cost quite a lot of money, there is clearly inadequate handling of the device, which can lead to damage. And these are the sort of things that we need to consider when we're developing instructions for reuse, to ensure that people understand that these sort of practices are not very good and can lead to significant risks, both to the device, to the patient, as well as to the staff. Now, the goals of device reprocessing in these and many other departments or parts of hospitals where the devices are being reused is to ensure safety. And there are 4 aspects to consider under this topic. Clearly, our first priority is safety for the patient. And this will include many different sorts of risks, be the microbial risks for infection as well as chemical risks for toxicity, or other complications from the use of that device on the patient, some examples of which I will show you in a few minutes. And then we have safety for the staff. So for example, how difficult is the device to disassemble prior to being cleaned? Are there parts of the device that could do them harm? Is there concerns about the how the cleaning is done to ensure that they're not exposed to risks during that cleaning step. And then the device, and this is clearly a great concern to us, because we can develop cleaning, disinfection, and sterilization methods and handling methods to be able to ensure the device is safely reprocessed. But if the device is damaged, that can often have a significant impact on patient health, and also could reduce the life of that device and expectation of what that device can do for the clinical staff or the surgical staff in particular. And then finally, we have safety for the environment, and this is indeed becoming quite an important concern, and something that we will also consider briefly later. So the risks of a device not being processed correctly can be quite significant. Now, first of all, as many of you work for companies that provide medical devices to facilities, there are clear commercial risks here. You've got the risks of reputation. So where your device has been implicated in an infection or a toxic outbreak within a hospital that then gets indicated as being due to lack of instructions for use or lack of good design of the device. This is not the sort of situation that we like to see. And that can also lead to legal and ethical issues within the company that can have quite a far-reaching impact. But some of the ones that we're more particularly concerned today would be microbiological, clearly. And these include the basic risk of contamination being transferred from one person or from the environment to another person at the wrong time in the wrong place. And do bear in mind that in hospitals today, which are often some of the best places in the world to pick up infections, due to the fact that we have lots of sick people that already have lots of infections in these facilities. And the opportunity for transfer to already compromised patients is quite high. So we even have to be a lot more diligent in these cases. And the risks that we pose of putting a device into a patient where it is normally sterile, an introduction of even low concentrations of microorganisms can lead to quite significant impacts and even death in some cases. The chemical risks we're concerned about are, for example, toxicity. So materials that can remain on the device can often still be there after the sterilization process. And so making sure that those residuals are not present is quite important. And that can vary depending on the design of the device and the materials that are used on the device. And that's become quite an important aspect to consider during validation. For some particular devices, there may be radiological risks as well, which is obviously a different type of chemical or physical risk. And this may also need to be considered depending on the individual device and its use. Finally, and many of you will know this if you have an engineering background, there are many mechanical and electrical risks. And so, for example, the opportunity to have staff electrocuted by a device that hasn't been well installated or has been damaged during use can be high, as well as mechanical risks. And here, for example, you see an example of stress cracking that can happen on a device due to multiple rounds of steam sterilization in this case. And of course, if that is not seen before use or may happen during use of the device, then that can lead to quite a significant patient implication. Now here are some more examples. The example on the left highlights the importance of tracking devices through the surgical procedure. It is often easy to lose one and forget that it maybe still remains embedded within the patient. And this, of course, is a patient consequence where the device now has to be retrieved from this patient. This reminds me to mention that the tracking of individual devices through a facility over time is becoming an important requirement, in particular in certain parts of the world, and it highlights the importance of using unique device identification for the future, such as GS1 codes, which I'm sure that many of you will be working on. There are also risks to the staff, as I said, on electrical and mechanical concerns, in particular for shocks that can pose an infection risk, and for patients where inadequate reprocessing can lead to canceled procedures and tissue damage. For example, here you have an example of a device that not only has lost a sharp edge, but obviously two of the tips have actually broken off And of course, when that is handed to a surgeon at the wrong time in the wrong place, that can have quite a significant implication. These are some other examples of material compatibility concerns, which we will talk a little bit later. But you will see examples here of adhesives breaking down, parts of devices being compromised due to the development of rust, and loss of color sometimes, which can lead to a lot of confusion surgically. Now, these are all sad examples, and they're quite clear examples of where the difficulty in the design of the device and how it is used, as well as the lack of attention to detail on various different processing steps, particularly cleaning, can lead to problems. These are all examples of devices that have been directly presented for surgical use, and you can see the concern. Visually, this should be obvious to us, but many times such devices are provided to the point of use in the operating room or other area, and sometimes we are lucky to ensure that they get rejected before they're used. Sometimes we are not. These are examples of toxicity risks due to inadequate processing, and are shown such as colitis from flexible endoscopy use due to the lack of rinsing of aldehyde chemistries that are used for disinfection. It's taken from the colon. Then we have an example of TAS, which is toxic anterior segment syndrome, a serious complication of eye surgery and the potential of orthopedic implant rejections. And due to the buildup of residuals that can occur either due to lack of cleaning or lack of rinsing, for example, where you would have cleaning chemistry residues remaining, or even residues from water that's used to rinse these devices over time. Of course, the buildup of those materials, and then when that enters into the patient and remains for a certain amount of time, can lead to an immune reaction, and that can actually lead to the rejection of the device, something that may not be noticed for some months. The greatest concern has certainly been particularly highlighted in recent years, which are infections. They're probably the most reported in the literature. And here's an example with flexible endoscopes and antibiotic-resistant E. coli and Klebsiella. And there has been a number of these types of outbreaks in recent years across the United States. These are quite significant because many of these antibiotic-resistant gram-negative strains are very difficult to treat in patients with only limited numbers of antibiotics available. So they have been a particular concern to healthcare facilities over the last year. And to see cases where these organisms have transferred on devices that have been inadequately reprocessed is certainly disappointing. Here's another example. These are with surgical devices, arthroscopic devices in particular. This organism that was transferred in this outbreak is Pseudomonas aeruginosa, which survived inadequate cleaning and disinfection and steam sterilization to lead to a patient's infection. Now, considering that this organism is rapidly inactivated by heat-based disinfection and sterilization, this outbreak is actually remarkable and shows that we cannot be complacent. It's clear that the difficulty in cleaning these devices was highlighted as being probably the leading cause of this outbreak, and highlights that even with the redundancies that we have from a safety point of view, that things can go wrong very easily. Certainly, the design of the device was also implicated as being an important factor in the outbreak. These are some examples of devices that are often considered problematic, or device features that are considered problematic. For example, here are 3 different generations of laparoscopic devices that have been improved from a cleaning perspective. So you can see the first design was very difficult to clean. The middle design, or second generation, was provided with a cleaning port. And then you have a third generation, which can be completely disassembled to allow for better cleaning and inspection of that device before sterilization. Good example. Now, other challenges that we have for cleaning will be obvious to you, such as dead ends, junctions or transitions in the device that can't be seen, the ease of disassembly, or maybe in many cases where the device cannot be disassembled, any narrow or inaccessible areas that can be different to access during cleaning, and valves for particular. Valves, for example, that can only be adequately cleaned by actuating them in an open and closed position to ensure flow during that process. Often instructions are also not clear, such as during the cleaning process, which types of chemicals can be used or maybe not used, which could damage the device. What are appropriate and inappropriate cleaning methods? Appropriate types of brushes, for example, and restrictions on that. And being able to immerse the device. So many devices we see in the field today are not allowed to be immersed, or parts of them can only be immersed, which makes cleaning very difficult. So it is easy to see that these things can lead to problems. The same is true for disinfection and sterilization. For example, with disinfection, what are the thermal disinfection requirements? Are there limits on the temperature that they can be exposed to this device? Is water quality an issue? So if you have high chlorine present in the water, is this going to impact the device? In most cases, the answer is yes, so there may need to be restrictions on that. Chemical disinfection can also be a concern. Certain types of aldehydes and alcohols are not recommended in certain markets, while in other markets they are widely used. So you need to consider both national as well as international requirements in these instructions. And some of the sterilization issues will include, can low or high temp sterilization methods be used, or both in many cases? Is there any difficulty in ensuring that the device can be dried correctly? And the use of extended steam sterilization cycles has created a concern, where we have found that there are sterilization cycles that are up to 30 minutes long, but these have never been approved as sterilization processes, for example, in the United States. So even though the instructions claim 30 minutes, they can't be run under a normal FDA-approved steam sterilization cycle. And when we look even closer into these cases, we find that the problem was not steam sterilization, the problem was steam conditioning, where conditioning cycles that are typically used in hospital sterilizers were not even tested, and they gave false results, which were then included in the instructions for use. Also, there are many types of requirements for certain markets, just like as I've highlighted for chemical disinfection, where you may have steam sterilization cycles that are just preferred in certain countries or approved in certain countries, like the FDA in the US, by the US FDA, as well as international, where you can have specific requirements. For example, in France, the use of a sterilization cycle at 134 degrees Celsius for 18 minutes is quite common. Now, a number of years ago, this became a great concern to healthcare facilities, to the manufacturers of devices, regulatory agencies, and standards bodies. So in the US, for example, AMI, and this is the publication they have, now AMI is the Association for the Advancement of Medical Instrumentation, convened a meeting with all these stakeholders, including the US FDA, to discuss this issue and highlighted 7 major concerns. These include things like how do we define cleaning and how is it shown? The importance of having standardized and clear instructions. The importance of human factors in those instructions. Ensuring that staff are adequately competent and trained and certified to be able to do these instructions correctly. But more importantly, to create a sense of urgency and understanding through the healthcare community of the importance of inadequately processing to patient safety. So these have led to new demands. And these demands will include greater attention by the regulators to detailed instructions. These instructions need to be validated much more than they were previously. And this will include both requirements here in the United States of America as well as internationally. We even know of certain conditions these days where new devices would be rejected from registration because of the lack of good detailed instructions for use. There are also legal risks, the risk to the company and to patients that we've mentioned earlier, as well as greater customer demands. These includes more detailed instructions, as well as making sure that they can meet the requirements for any accreditation organizations such as Drug Commission, which is a great focus for those accreditation organizations today. So where to start? Well, the first is to look at the range of guidelines and standards that need to be met, both by the manufacturer and also by the customer. These are just some of the many agencies, and this is just for the United States alone, that are involved. And there is a lot of things to consider in each and every one of these. And it's our job collectively to sometimes harmonize these into instructions for use, as well as to make sure that they are followed correctly within healthcare facilities. It's not easy for a healthcare facility. This can be very confusing and can include many aspects. It can have impacts, for example, on the suppliers, where we have to be able to meet various different guidelines and standards to ensure that we can legally market a product in, for example, the United States of America. And then there are various different organizations that tell the health or advise the healthcare facilities on how to do this on the left of the screen. And then you've got the impact of CMS or the insurance companies ensuring that those facilities are being audited and accredited to ensure that that can be safely amalgamated for the patient. Not easy. And considering these many differences between regulations, standards, and guidelines, it can be quite difficult to plow through, not just for ourselves, but also for our customers. These are examples of specific healthcare facility guidelines, and they include standards such as ST79, which is more specific for steam sterilization, as well as ST58 for chemical sterilization and high-level disinfection, as well as the AORN recommended practices. These will all recommend various different aspects of cleaning, disinfection, rinsing, sterilization, storage. We have similar examples, for example, in Australia and New Zealand, and this guidance document on reprocessing is actually currently being revised. We have a new series of standards just released from Canada, which also is looking for the same sort of information. And this is not only across Canada, but also some specific requirements in different provinces. And then final example I've given on this is for the United Kingdom, who have just released a whole new set of management guidelines on the decontamination of surgical instruments used in acute care. Specifically for the manufacturers, these may be things that you're more familiar with, and in particular regarding reprocessing. So the major goal here is to have clear instructions for use. And the key standard in this case is ISO 17664, which is information to be provided by the manufacturer for reprocessing of re-sterilizable devices. We have revisions ongoing with this, and we'll review this a little bit later on to include various different aspects of cleaning, disinfection, sterilization, and biocompatibility. And then we have specific guidelines. For example, Health Canada have a guidance document of really reiterating the same information. The A&E TRR 12, which has been in place for a number of years. And then we've even had a great focus on this from the FDA in 2011, which really focused our attention on this area. And many of you may be familiar with this draft guidance for industry and FDA staff regarding reprocessing of devices and the validation of methods and labeling requirements to include instructions for use. This has become essential for 510K applications in the United States. And I've already mentioned that if this is not done correctly, this can lead to rejection by the FDA. So let's maybe now look at some of the top considerations in this area as we move through the reprocessing cycle. And for that, I will now hand you over to my colleague, Beth, who will proceed with the rest of the presentation.
Thank you, Jerry. From our experience, there are 7 common and important themes when considering reprocessing reusable medical devices. These are compliance to a particular standard, that being ISO 17664, and the associated issues specific to risk analysis, material compatibility, cleaning efficacy, antimicrobial efficacy, toxicity and safety, and human factors. These 7 items are critical to the development of acceptable instructions for use, both nationally and internationally. We will discuss each one of these briefly in the following slides. The ISO standard 17664 provides valuable guidance, particularly for information to be provided in the instructions for use of your reusable medical device. The document title implies a focus on sterilization, but it provides requirements for the entire reprocessing process and provides an example IFU with all the appropriate sections from point of use through sterilization and storage. Development of instructions should also include applicable regulatory requirements, commercial requirements, and U.S. and international requirements. This is an example of cleaning instructions. It is important that instructions for use provide information on what to do, which is provided in this example, but not to any great detail. It is also important to provide information on what not to do. This photo provides some information to the user of the correct way to wash a baby and the incorrect way. You may identify the use of a neutral chemistry under the right conditions, but if your device or baby cannot withstand a high pH cleaning chemistry, a cautionary statement should be included to ensure any risks to the safety, functionality, or life of the device are minimized. This is a better example of instructions, with details being provided to meet customer and regulatory expectations. Just look at compliance to ISO 17664 on cleaning alone. This is taken directly from the standard and specifies requirements for preparing the device for cleaning. For example, the importance of disassembly of the device and tools or steps that are needed for cleaning. The standard also requires validation of cleaning methods. Both a manual and an automated cleaning process are to be provided in the instructions for use. Note that in many countries like the U.S., manual cleaning is often preferred. However, in other parts of the world, such as some specific European countries, automation is often the customer requirement. If the device cannot withstand automated washing, a caution should be included in the instructions, warning against automated washing. The cleaning information required can include types of cleaning chemistries, water quality, temperature restrictions, etc. Remember, these methods should be validated by the manufacturer, and this often requires close collaboration with the equipment manufacturer and validation test laboratories. Here is another example from Health Canada on the level of detail required for cleaning instructions for use. This approach allows for recommendations and cautions at each step of the process, and clear guidance regarding the specific information required from the manufacturer to perform each step of the process. As Jerry mentioned previously, the FDA have recently become concerned about instructions for use and have published draft guidance for processing, reprocessing reusable medical devices in May of 2011. This guidance highlights 7 criteria for medical device reprocessing. These include correct and detailed labeling that reflects the intended use of the device, a change in perspective on the details for cleaning and how that is validated, requirements for statements of disinfection and/or sterilization based on the Spalding classification, clear but also practically and technically feasible instructions, the use of equipment and accessories that are legally marketed in the intended country. This will include defined sterilizers, washer disinfectors, cleaning chemistries, and high-level disinfectants, among others. The instructions should be comprehensive and include all steps from point-of-use cleaning to next patient use and company contact information to address questions. And finally, the instructions should be understandable, written in clear, grammatically correct English or other applicable languages, be legible and in logical order. There's a lot to consider, and the expectations have never been higher from both your customers and the regulatory agencies. A good way to approach processing from the start is by risk analysis. Risk analysis and management is critical to ensure patient and clinical staff safety. It is intended to be applied to all stages of the life cycle of a medical device, including risks associated with reprocessing. As part of the risk analysis activities, the design and use of the device should be considered both from the point of view of the physician using the device and also the personnel reprocessing the device. Risks to consider may include mechanical, electrical, chemical, or biological hazards resulting from reprocessing steps. These risks should ideally be considered early in the development of a device, rather than at the end of the development cycle. Safety risks need to be balanced. These will include safety for the patient and the staff performing reprocessing, such as microbial and chemical toxicity risks, safety for the device or materials compatibility, and finally, even for the environment. Many of you may already be involved with sustainability projects for companies and healthcare facilities to reduce environmental impacts. Let's consider materials compatibility. When designing a reusable medical device, materials should be selected with reprocessing in mind. Poor selection of materials could result in the issues shown in the photos, such as adhesive degradation, rust, or loss of color on anodized components. The design and construction of the device should be tested through the life expectancy to confirm integrity and functionality of the device over its use life. When designing fatigue studies, consider subjecting devices to worst-case scenarios, which may include high alkaline cleaning chemistries, extended heat, disinfection or sterilization cycles, or chemical disinfection or sterilization. These studies may need to be performed in one or more of these combinations. If using materials with known chemical limitations, be sure to include these in the instructions for use. For example, the device may not be compatible with chlorine-based chemistries and should result in a statement in the instructions to avoid these types of solutions. As mentioned earlier, regulatory agencies such as the FDA also recommend in their draft guidance that the manufacturer provide lifetime expectations and/or inspection criteria to indicate the device end of life. Let's change now and talk about one of the most discussed requirements for device reprocessing, the importance of cleaning. In order to be able to clean reusable medical devices, we need to have a better understanding of what is present on the device after clinical use. So let's take a closer look. This is a summary from a new study, soon to be published, looking at contamination levels on devices directly following clinical use. Devices included surgical devices from a variety of procedures and flexible devices used in colonoscopy. The results of the study show that protein is a major contributor to soil remaining on all devices, but you will also note that microbial contamination was much more significant on colonoscopes. These results are not unexpected when considering most surgical instruments are used in sterile areas of the body, while flexible colonoscopes enter the colon. Other soil indicators, such as total organic carbon, TOC, and hemoglobin may also be useful. This will be important to consider when designing studies to validate that cleaning is effective. So let's consider cleaning further. When determining appropriate cleaning steps, national and international requirements should be considered in accordance with the region in which your device will be sold. Test methods for validation are developed from these considerations and will include rationale for selection of the test soil and how the device will be soiled to simulate clinical use. Cleaning endpoints need to consider what is seen and not seen, as demonstrated by the cat. These requirements will include visual inspection, but also biochemical assays. Acceptable biochemical endpoints are identified here, such as for protein, TOC, and hemoglobin. These levels are the basis for a new standard under development for cleaning efficacy by ISO and includes FDA recommendations. In addition, devices must be shown to be safe or non-toxic following cleaning as defined in the ISO 10993 biocompatibility standards. It is important to note that there are a wide range of cleaning chemistries and equipment, such as ultrasonics and washer disinfectors, used worldwide that may need to be considered in the development of instructions. An important step in processing is to reduce or completely remove microbial risks that could lead to contamination or infection. This slide shows the different levels of device classification according to the Spalding classification, and some examples of the different devices you may find in each category. You will see that cleaning is always recommended, but this may need to be followed by disinfection or sterilization. It is important to note that some interpretation of this classification system is needed. Devices can belong in different categories depending on their clinical use. For example, a flexible endoscope is considered semi-critical when it is used in a procedure where it only comes in contact with mucous membranes. But if that same flexible endoscope is now used in a different procedure where it comes in contact with blood, it is now considered critical. Also, non-critical devices present a low risk, but not no risk. Many of these devices can be environmental sources of healthcare-acquired infections, and recently the FDA, in particular, has raised expectations for reprocessing instructions for these types of devices. Many of you may think that the risk of infection or other complications from reusable devices are very low. Consider these statistics briefly. 10% of patients in healthcare facilities acquire a healthcare-associated infection, and these numbers will vary considerably worldwide. It is estimated that 3% of patients will acquire a surgical site infection despite the many precautions, and these estimates also vary to include many types of bacteria, fungi, and viruses. Recent publications have highlighted lapses in device reprocessing as being the cause in many of these cases. There are also risks associated with other devices, such as semi-critical flexible endoscopes being the leading cause of device-related infections, and even non-critical devices as being a source of microbial contamination to patients. This chart shows a range of microorganisms and their relative resistance to inactivation. It is generally considered that bacterial spores show the greatest resistance to inactivation, while enveloped viruses such as HIV or the much-discussed concern of the day, Ebola, are more easy to kill. The expectations for the various inactivation methods, such as sterilization and the different levels of disinfection, are shown and need to be considered during validation. Our next consideration is toxicity, such as cytotoxicity, irritation, sensitization, and even carcinogenicity. There has been an increased emphasis on this issue as an important consideration for the design of the reusable medical device. Not only are materials of construction and intended use to be considered in the design of reusable medical devices, risks associated with the reuse of the device must also be considered. Various cleaning chemistries and physical methods of soil removal can affect the device materials or build up on the devices over time. Short and long-term risks include complications during eye surgery and rejection of orthopedic implants. Other safety considerations for reusable medical device safety will include protection from electrical malfunction and cuts or punctures from sharps. Human factors engineering is a topic that most medical device manufacturers know and understand. It is important to consider this during the development of instructions for use for reprocessing. For example, the FDA draft guidance for reprocessing includes a section on human factors. The recommendation is to develop consistent reprocessing instructions across each manufacturer's product line. These will include using the same layout for all device types to improve comprehension and adherence to the instructions. Instructions should take into consideration the processing environment, personal protective equipment, the weight of an instrument set, disassembly and reassembly requirements, inspection, the correct positioning of the device for cleaning, disinfection, sterilization, and drying, the extent of manual cleaning, and verification of cleanliness. It is recommended that manufacturers independently verify the written instructions to ensure they can be successfully followed. In summary, device processing, reprocessing is an important design criterion for reusable medical devices. There are increasing customer needs for accurate instructions for use or reuse. The important 7 topics to remember when developing a reusable medical device and its instructions for use are compliance to ISO 17664, performing a risk analysis, the importance of device safety for material compatibility, cleaning efficacy, antimicrobial efficacy, toxicity, and human factors. These considerations should lead to a well-designed device and associated instructions written in clear, simple language and validated as part of design control. Biotest Laboratories supports manufacturers of reusable devices in design, final development, and validation testing of their devices and instructions for use. Their services include reusable device testing, microbial testing and validation of processes and sterilization, steam sterilization, contract manufacturing and packaging, clean room and clean bench certification, and external training and consulting. Biotest Laboratories is part of the larger STERIS Corporation, a leading provider of cleaning, disinfection, and sterilization equipment, chemicals, and service for infection prevention and contamination control. For further information on processing or reprocessing of reusable medical devices and the validation of instructions for use, please contact us directly or contact Biotest Laboratories to be directed to personnel specializing in validation. This concludes our presentation. Next, we would like to take a few questions. If you haven't already done so, please type in your questions and provide your contact information within the field provided on your screen.
Okay, so we seem to have one question, which is markers for FDA cleaning validation usually includes protein and hemoglobin. Does the CE mark requirements also include just 2 markers? There is actually no actual requirement for that, but today I should point out, because the major requirement seems to be around protein. But the trend that I can tell you about for cleaning validations in particular is to use 2 markers, with the 2 most prominent ones that we hear discussed are, of course, protein and total organic carbon. We have a next question, which is, can cross-linked poly or E-poly be sterilized more than once? Well, I'd like to suggest that maybe you'd be the better person to answer that question, because it really does depend on the validation. So if you can show that it can be sterilized once and has more times and has no impact on whatever safety requirements that you have regarding that, then it should be fine. The other thing I would suggest to you, Mark, on that is that you consider the sterilization method that you're using, because indeed the answer may be yes for some sterilization methods and no for others. So that would be something that you would investigate during, hopefully, the design controls of the device. A question from Cecile, thank you for the question, which is, how do we ensure that all parts of the device remain together? Well, we don't. This is something that needs to be managed by the facility. Now, there are 2 things to consider here. First of all, can you design the device in such a way that could keep those parts together, but not compromise access to the device. And that's the sort of thing I think that we're trying to encourage in many ways during the design. And so many of these issues that we see with cleaning, disinfection, and sterilization could have been addressed earlier in the design phase if they would have been considered by the engineers designing them. And so in many cases, it's now becoming a requirement that reprocessing is considered much earlier during the design process rather than at a later stage, when sometimes it's too late to change the design. But I should point out that, of course, the major requirement is to ensure that you've got the right instructions for use, independent of what that device is, assembled or unassembled, and to recommend to healthcare facilities how they keep that together. If they don't follow those instructions, then I really have no other advice to offer on that. I have a question from Eric. Eric, pleasure to see you, or at least hear you again. So what's the number of the standard in the development for protein and hemoglobin levels, and how are these levels determined? Well, these, first of all, the standard that is under development right now is being proposed to be ISO 15883 Part 5. Now, many of you will know that that already exists, but it doesn't exist in its standard format. It exists as a technical specification which provides advice on validation of cleaning. And so we are currently working on a revision of that, which will be upgraded, we hope, to a standard which will include these requirements. Now, many of you will also know that that is a standard defined for washer disinfector manufacturers, not actually for instruments that would go into the body. But we believe that that will become the standard for cleaning efficacy across the world, not just for wash disinfectors, but for any device that will require to be cleaned to a safe level. And how were the levels determined? They were determined based on the review of the literature and levels of our understanding, at least today, on what levels are found on devices following vigorous cleaning, or also with consideration of what levels may potentially be a source of risk. They are not perfect, but in many cases they are where the science is today in this level. And they're also reflecting the regulatory requirements and expectations that we are hearing both in the United States as well as internationally. Okay, and then the next question we have from Cecile, I'm sorry I missed that question, which are, how are reusable devices prepared for reuse after a contamination event, such as a flood during Hurricane Katrina, I'm sure you mean, and burst pipes or other leaks onto the equipment? Well, within facilities today, they would follow the same requirements as you would normally follow. So, for example, we have what we call standard or universal precautions of care. So the same cleaning steps, the same disinfection steps, and the same sterilization steps should be provided, because as you well imagine, and we have this question on a daily basis regarding Ebola today, what do we need to do different? You don't need to do anything different in many cases. You need to ensure that you're following the standard precautions that have been applied. In many cases, we don't know what's on the device when it comes out of a patient, and it may include a variety of different types of infections or infectious agents. And we have a question from Natalia, which will be our last question today, which is that you indicated that the limit of protein is 6.4 micrograms per centimeter squared. This is outdated. Amy, TIR 30 2011 sets the limit for protein at 6.12. Natalia, I'm not going to argue with you on a number of points on the microgram scale. As you can see, they're in approximately the same range. The number that I have given you is the current number that is written into most of the guidelines and standards that I see today. And I'm happy to go back and check TIR 30 2012. But as you can see, they are approximately in the same range. And I don't think we would get down to that level of specificity. So on that, I will hand it back to our convener.
We still have a few questions remaining, and in consideration of everyone's time, we're going to wrap up the webinar right now. But as we mentioned in the beginning, for those questions that we weren't able to address in the time allotted, Beth and Jerry will follow up with everyone in email to make sure we address those questions. And of course, if you have any other questions that might come up, you have the contact information right there on the screen, so please feel free to contact us for any assistance. Thank you everyone for joining our webinar today. We certainly appreciate you taking the time to learn with us, and we hope you found this session beneficial. As a reminder, we'll email a link to download a recorded webinar tomorrow. Thanks again for joining us today. This concludes today's webinar.